So guys, we'll quickly look at the production of matured red blood cells. And looking at the production of matured red blood cells, that's where we get to look at all those stages, okay? From the pronormoblast, the normoblast, erythroblast, basophilic, polychromatic. It goes down like that, all right? So we'll be looking at it, and we'll be looking at each of those stages, their characteristics. Because this is a very good essay question in anything blood physiology or hematology, okay? There's a 50% chance that you be asked this, okay? Once they actually treated blood physiology with you, they'll always ask you this, okay? Let's look at the production of what mature red blood cells. That's basically what mature, the different stages that are included before you have this erythrocyte, or you could say the mature red blood cells being formed, okay? So from the introduction right now, we see that erythropoiesis is the process of what red blood cell formation, which usually occurs in the bone marrow and involves the bone forming cells and tissues of the body. Okay. Now differentiation for the hematopoietic stem cells. Stem cells differentiate to form multipotential progenitor cells. The erythrocells are formed by the granulocyte, erythrocyte, monocyte, and melloblast multi-lineage. Okay, so they have like a big lineage that has the capacity of forming a granulocyte, has the capacity of forming erythrocyte, okay, or erythrocyte progenitor cells, have the capacity of forming melloblast, have the capacity of forming what monocytes. Okay, so now the erythrocells, those are the what parent cells for erythropoiesis, are formed from this multi-lineage. Right. So now erythroid maturation and differentiation is dependent on the microenvironment, which is provided by the stroma cells of the bone marrow. This is like the environment that is what making the bone marrow conducive for what red cell formation. That's a what hematopoietic induction microenvironment. I have a different video on that. All right. Now, this environment is formed by stroma cells, and these cells include fibroblasts, reticular cells, adipocyte, macrophages, and endothelial cells. Okay. So now we say that what there are also immune regulatory cells, which include monocyte, macrophages, lymphocyte, which contribute to what local cytotoxicity. Right. So now after the erythroid program has been certified, the final phase of erythropoiesis involves maturation of what erythroid progenitor cell. Okay, so you can see it. It's a very nice image for everything. Okay, and my mnemonics for this is what I call the PB poem. All right, so these cells include pronormoblasts. All right, P is what? P is pronormoblast. My B is what? Basophilic normoblast. P is what? Polychromatic normoblast. O is what? Orthochromatic normoblast. The R is what? Reticulocyte. The M is what? Matured erythrocyte. Okay, so PB poem. Know this, then we can move. Okay, so moving right now, guys. So the first set is what? You say proerythroblast or pronormoblast. It's the same thing. Okay, so talking about the characteristics of what red blood cells that are at this maturation stage, so that this corresponds to the colony forming unit erythroid. All right, the red blood cells here are relatively large and are between what 12 to 20 microns in diameter. All right, they have a non-granular deep blue cytoplasm. They have a large nucleus that occupies about what 3/4 of the cell, almost occupying all the cell. Okay, so you see that what now, you guys see that as we continue talking about this story, we'll always talk about this nucleus, how the nucleus is decreasing in size till when the nucleus is sent out and the real mature red blood cells doesn't have a nucleus. Okay, so their cytoplasm contains numerous ribosomes, centrals, permanent Golgi apparatus, few strands of what ER, several mitochondria. Okay, and these ones now will differentiate to produce what basophilic normoblast. Now let me give you guys a hack. Anytime we're talking about the next stage, you can see that what The basophilic normoblasts are formed from the pronormoblast. Okay, I can see that basophilic normoblasts give rise to polychromatic normoblasts. That's 2 points for free just by knowing PB poem. Okay, once you know PB poem, you know that which one is which one is before which one is after which. Okay, so the characteristics of this basophilic normoblast, these ones are smaller than the pronormoblast. All right, that means they decrease to about what, 10 to 12 microns in diameter. All right, 10 to 16, sorry. This one was 12 to 20. All right, you have a deep blue cytoplasm. The nucleus occupies large portions of the cells, but the nucleus is now reducing in size than the pronormoblast. Those are 3 points. They have a coarse reticular chromatin pattern adjacent nuclear membrane. They can also differentiate to produce what? Polychromatic normoblast. Okay, see it, right? Be from the basophilic erythroblast or the basophilic normoblast right now. Okay, most of them have 2 names, right? They can differentiate to form the polychromatic erythroblast, also known as a polychromatic normoblast. Okay, so moving to the polychromatic normoblast right now, we see that they have a blue-gray cytoplasm. There's an increased development of a pinky cytoplasm. They are smaller in size now. That's between 10 to 12 microns in diameter. This one was 10 to 16. All right. They divide to form orthochromatic normoblasts. That's a hack. Always talk about what forms them, what they will form. 2 free marks. Okay. Then moving to the orthochromatic normoblast right now. That's what we got here. We say that this one, they have a pinky cytoplasm, of course. Hemoglobin is being formed right now. They are non-dividing cells. Okay. Now, as the cell proceeds through the terminal stage of differentiation, the nucleus condenses further until it is finally extruded in serum to form reticulocyte. All right. So it's basically at the orthochromatic normoblast stage that you find the nucleus being given out. Okay. Then next we have the reticulocyte. This is just the stage before the matured erythrocyte. This one has no nucleus, few mitochondria, few ribosomes. The cytoplasm stains predominantly pink because of the high concentration of what? Hemoglobin. Cytoplasm has a grayish tint due to the presence of what? Ribosomes. There's a continuous synthesis of what? Hemoglobin, right? Between 24 to 28 hours after release from bone, right? Because the reticulocytes are released from bone. So now these ones are 20% larger than the matured red blood cells that we have. Okay, so we are at the reticulocyte. Now find any of these pictures and draw them out. You mustn't just stick to one scheme, right? Just stick to one drawing, okay, and perfect it. Okay, so all of them are same drawing in different ways. So the mature red blood cells now, these are the final products of erythropoiesis. The cell is circular, biconcave and have a diameter of about what? 8.5 microns. Okay, so that's just simply what production of matured red blood cells. Stages in the production of matured red blood cells. Is it 5 now?